Hi guys, this is Mrs. Foy, and this is part 1 of the AP Biology lecture on photosynthesis. And I am so excited to tell you about this process because it is amazing. It's just a miracle of evolution. So this chemical process feeds the biosphere, right? So the great majority of life on Earth gets its energy from glucose. And glucose, of course, is the product of photosynthesis. So here we go. We're going to dive right in. So when you see a diagram like this, you should be able to write the equation of photosynthesis, and then you should be able to write the equation of cellular respiration. So let's go first with what we do, okay? What we as heterotrophs do. Cellular respiration is going to be glucose, right? Plus oxygen, and that's going to yield carbon dioxide, water, and ATP energy, right? So we're going to get this energy that's going to run all of our endergonic reactions as cells. And of course, all living things do this, right? So plants and animals do this. The great majority of living things do cellular respiration, except a few crazy bacteria that get their energy from inorganic means. But what drives this? What drives cellular respiration? What has to come first is photosynthesis. So photosynthesis is the reciprocal reaction, right? So we're going to take carbon dioxide and water, and we're going to take the energy from the sun, because this reaction is an endergonic reaction. It's a positive delta G. And we're going to make glucose and oxygen as a waste product. So what we have here is we have a recycling of all of these nutrients, a recycling of all of these atoms except the energy, right? We first law of thermodynamics, right? Energy can be transformed but not created or destroyed. And every time that we transfer that energy, we're going to lose some in the transfer. So we have CO2 as a reactant in photosynthesis. CO2 is a product in cellular respiration. Water is a reactant in photosynthesis. Water as a product in cellular respiration. Glucose as a product in cellular respiration, glucose as a reactant glucose as a product in photosynthesis, and glucose as a reactant in cellular respiration, and oxygen as a product and oxygen as a reactant in cellular respiration. So big picture here, I have this light energy coming in, and this is a good time to talk about these little packages of light that we call a photon. So think of if you haven't had physics yet or chemistry, it's easy to just think about a photon as a package of light energy, all right? And so that's hitting these leaves. We have organelles called chloroplasts that are going to be where a lot of the action is. We have carbon dioxide as a reactant. It needs to come into the leaves because this is where we're going to have photosynthesis occurring. And right away, we're going to see oxygen being given off as a waste product. The water comes in from the roots, as we know, and we're going to talk about how that gets up to the leaves. And water is going to be our source of hydrogen here. And then we are going to make glucose. We're going to make sugar. And that sugar is going to be burned immediately in the plant's mitochondria, or it could be stored. It could also be stored. If it's not used as the plant's fuel, it could be stored, and then some heterotroph like you or me eats it. So this is a review of cellular respiration. You should have this so entrenched in your brain that you are not going to get confused when you look at photosynthesis, because there are some similarities, and it's almost kind of like looking in a mirror. Okay, so we have glycolysis first. We know that happens in the cytosol of each cell. And then we know that we have it happening in the mitochondria. We're going to take pyruvic acid. These are 2 3-carbon molecules. It's going to go through the link reaction. That's the first place we have CO2 coming off. And then it's going to go into the Krebs cycle, or the citric acid cycle. And in both of these processes, in glycolysis and in Krebs cycle, I am going to generate some high energy electrons that came from the glucose, right? And they're going to be carried on these carrier molecules NAD, and that is going to be reduced to form NADH. And then all of these electrons on the NADH and the FADH2 are going to come into the electron transport chain, and through a process called oxidative phosphorylation, which includes chemiosmosis, right? So we learned about these hydrogen ions that are going to be pumped in the intermembrane space of the mitochondria between the cristae and the outer membrane. And then we're going to see that we have ATP synthase. We have ATP synthase that allows those hydrogens to go back down their concentration gradient, and I make a whole bunch of ATP via oxidative phosphorylation. Now, I do make some ATPs, remember, through something called substrate level phosphorylation. So I make a few ATPs in glycolysis and a few ATPs in Krebs cycle, but the big gangbuster amount I make in oxidative phosphorylation. And of course, oxygen is my final electron acceptor, and then I have hydrogen protons that are just around, and that's going to give me water, and that's my final product. What we're going to see in photosynthesis, guys, is going to be basically the mirror opposite. We are going to see an electron transport system that's going to be very similar to what we saw in the mitochondria in the light reaction. And instead of it happening on the membranes of mitochondria, we're going to see it on the membranes of these organelles, the chloroplast, and inside the chloroplast we have these structures called thylakoids. And we're going to see the electron transport chain, a similar one to what we saw in respiration. We're going to see that in the first part. So remember how cellular respiration had 3 parts, right? It had glycolysis, Krebs cycle, and the electron transport chain. Photosynthesis has 2 parts. It has Act 1, which we call the light reaction, or sometimes we call it the light independent reaction because the light dependent reaction because it has to have light. And then the second part of photosynthesis, Act 2, is called the Calvin cycle. Now you might say, wow, Mrs. Foy, there was a Krebs cycle in cellular respiration. You are correct. So you can't get these confused, all right? The Calvin cycle is named after a guy named Calvin who worked out the chemistry of that, so we named it after him. But this is also called the light independent reaction. The light independent reaction. Sometimes we also call it the dark reaction, even though that's kind of a misnomer because it doesn't have to happen in the dark, it just doesn't need light. But this is going to be a cycle that's going to look familiar in a way to what you saw in the Calvin cycle. And at the end of this, we're going to make oxygen, at the end of the second process, we're going to make sugar. And at the end of the first process, we're going to make oxygen. And then we also see this ADP plus P going in here. And then we see something a little bit different. We see this NADPH. So that's not a typo. It's not NADH. It's NADPH, which is kind of a cousin electron carrier to NAD. All right, so here we go. So this is going to look very confusing to you right now. I want you to go back and look at this slide when you have digested all of this lecture. And you should be able to draw this, guys. You should know these two processes so well that you can draw this diagram and you know where every reactant and every product is, and you know each of the steps. All right? So here we go. Cellular respiration, right? Cellular respiration takes in oxygen, gives off CO2, gives off water, and takes in glucose. And there's our three steps: glycolysis, Krebs cycle, an electron transport chain. Here's our electron carriers, NADH. Photosynthesis is going to start with the input of the sun and water, and it's going to make at the end some ATPs, and also it's going to carry some high energy electrons on the NADPH. Now, these high energy electrons are going to come from a special place, and I'll show you that in just a minute. But the ATP and this NADPH, right? So these high energy electrons are the input along with carbon dioxide to make glucose. All right, that's going to be super important in the light independent reaction. So the bottom line is this: plants need carbon dioxide and water and the sunlight. That's what they need to make glucose. But this is an endergonic reaction. And so I'm going to need ATPs and I'm going to need some electrons. Now remember, in biological processes that are always done around water, you can tell where the electrons go by watching where the hydrogens show up, because the electrons, of course, are negative, and there's going to be these hydrogen protons around everywhere. So if you want to watch what's oxidized and what's reduced, watch the hydrogens. Watch where they appear. It's a little cheat about trying to figure out what's oxidized and what's reduced. So what you just need to just think about for a minute is, like, look at a tree. All right, look at the mass of that tree. Super heavy, right? Where did that come from? Well, it came from literally thin air, right? It came out of carbon dioxide and water. And with the energy from the sun, the plant makes its own building materials, including glucose, which is used to power its metabolic activity in the mitochondria. So we talk about photoautotrophs. Who are the photoautotrophs? Well, basically plants, algae. These are multicellular algae. We have unicellular protists. We saw some of these under the microscope. We have cyanobacteria, and these little guys are super important because they are the ones that built up most of the oxygen in our atmosphere over billions of years. So thank you, cyanobacteria. And then we have some kind of strange purple sulfur bacteria that also would be considered photoautotrophs. So where is this happening? All right, so let's look at a leaf. All right, so we're going to blow up a leaf. We're going to take a little piece of a leaf and we're going to blow up what the leaf looks like. So we need to understand the anatomy, guys, to be able to understand how photosynthesis works. So the first thing I want to point out to you are these little structures called stomata. And stomata are kind of like plant nostrils. All right, it's kind of like the plant breathing. So in these little openings in the leaf, you have carbon dioxide going in and oxygen being released. Okay, so it's just kind of the opposite of like what our nose would do. We also have these cells in what we call the mesophyll. And the mesophyll area is where we have cells that are going to actually do photosynthesis. So here is a mesophyll cell. And you can see those little green jelly bean things that you see there are the organelles, the chloroplasts, and that cell is chocked full of them. So you should be able to recognize that that is a cell that's going to do some photosynthesis. And so here is one chloroplast, all right? So here is a transmission electron microscope picture. And so I can see it's kind of a cross section that is giving me what the organelle looks like. And in the cartoon, I could see it even better. So I have these little discs called thylakoids. And I think of a thylakoid as one of my favorite candies. There's a candy called a peppermint patty. And they used to be made in York, PA, which is where we are from. These have chocolate. If you know this, they have chocolate on the outside if you break them open, and they have white kind of peppermint stuff on the inside. So I think of these thylakoids as a peppermint patty. A stack of them is called a granum, which is singular. A bunch of stacks would be called a grana. A grana would be multiple stacks. And then, of course, I have a double membrane, right? I have an outer membrane and an inner membrane. That's the inner membrane space. Sorry, I have an inner membrane. And, of course, we think that it has a double membrane because we think that this was once an autotroph that began to live in an endosymbiotic relationship. And, by the way, chloroplasts also have their own genetic material as well. So now we're looking at a chloroplast, right? So this would be one chloroplast that we would see in a cell. And here is another structure that I need to tell you about, the stroma. And this is some words that you're going to get confused. The stomata is the plant nostril. Okay, so you would see that when you look at the leaf itself. The stroma is the jelly-like substance that's inside the chloroplast. Okay, and we're going to see that a certain part of the photosynthesis happens in the stroma. Actually, this is where you're going to have Act 2 happening. And Act 1 of photosynthesis is going to happen on the membrane of the thylakoid, right? And again, look at all this membrane surface area. You have all of these little stacks, right? That gives me an idea that something very important is going to happen on those membranes. This is a great picture. I love this picture. So this is a picture of a leaf, all right, a cartoon of a leaf. And my outer substance that I have here is a cuticle. So we can think of that as kind of the covering of the leaf. A lot of times it's a waterproofing kind of material. And then we have the upper epidermis, like we have an epidermis, right? It's kind of like skin. And then we have these palisade mesophyll cells. And here we have some spongy mesophyll cells. So these mesophyll cells are where photosynthesis occurs. And you can see these little green dots here are the chloroplasts. So obviously those must be photosynthesizing. And then I have a stomata, right? So here's the plant nostril that's there. And on either side of these plant nostrils, the stomata, we have these cells called guard cells. And we're going to learn about how those can open and shut the stomata, right? So they can, by swelling up and decreasing their turgor, they can open and shut this area here. The other thing I want to point out is that this is what we call a vein, all right? And this is where materials are transported, just like in a animal vein or artery. And this is made up of two different kinds of tissue, xylem and phloem. All right, xylem and phloem, which we'll talk about. And there's some other cells that are very specialized in photosynthesis called bundle sheath cells. And just kind of tuck those away in your memory because we're going to be talking about those later. So I want to talk just a minute about organs and tissues in plants. All right, so the new AP curriculum doesn't talk about plants very much, unfortunately. But I just want to take this time to talk about organs in a plant. So you might not think that a plant has organs. It doesn't have a heart, doesn't have a kidney, but it does have flowers, or most plants, angiosperms, have flowers. It has leaves, it has stems, and it has roots. And these are the organs of a plant. We also have tissues. We also have different tissues in a plant. We have dermal tissue that's on the outside of the plant. We have vascular tissue, which is going to be the tissue that's going to transport. And we have two different types. We have phloem and xylem. And xylem transports water. And you're going to remember this because you're going to remember W, X, Y, Z. So this is going to help you remember that xylem transports water. And phloem, you can think of it phonetically. Phloem transports food. All right. And then the other tissues are what we call ground tissue or fundamental tissue. And that's just going to be tissue that's not dermal or vascular. So in a xylem vessel, and by the way, a xylem and phloem would be found together in a leaf vein. Okay. So xylem is going to transport water. We need water for photosynthesis, and this is always going to come up from the root. Okay, so plants are going to absorb water from their roots, and so by a certain types of characteristics of water. So we're going to learn. You've already learned about adhesion. And you've learned about cohesion. And then we're going to learn about something called evapotranspiration. Evapotranspiration, which is kind of like plants sweating water. That is going to set up kind of a vacuum system that's going to pull that water up. And so water goes up, and the water is going to be delivered to the leaves. Okay, and so that's where photosynthesis occurs. The phloem vessels are going to transport food, and AKA this is going to be sugars, right? It's going to transport sugars up and down. So the sugars are produced in the leaf, but the sugars are going to go down to the roots, but also up and down as well. And so we're going to see these sugar molecules being transported up and down. There is some really cool transport that happens with sugar. We have the source of the sugar, as I said, being in the leaf. Now this says sucrose. Sucrose, of course, is a disaccharide. So that would be two glucose molecules together. And what happens is there's a special type of junction between something called a companion cell. And then we have these sieve tube elements in the phloem that's going to allow for the transport of sugar. Some of the sugar goes down and is stored in the roots. All right. So when you think about a carrot, right? Carrots really taste pretty sweet, right? And those sugars and starches are actually stored there, but they were made from the sugar that was photosynthesized in the leaves of the carrot. So let's just look at comparing these two very important organelles, mitochondria and chloroplast. I would always suggest that you start with what you know better. So let's start with the mitochondria. I have glucose, I break it into pyruvate, I go through the link reaction and make acetyl-CoA. That gets fed into Krebs cycle, carbon dioxide comes off, I get a whole bunch of NADHs. I also make some NADHs up here in glycolysis, right? And these are carrying high energy electrons that go into oxidative phosphorylation, the electron transport chain, and it's going to make me a whole bunch of ATP. Oxygen is needed as the final electron acceptor. And when oxygen reacts with the hydrogens that are around in the electron, it makes water. Now let's look at what's happening in the chloroplast. This time I want you to start down here, all right? So what happens first is that there is an electron transporting system that we call the photosystems. It's going to transfer electrons in a hot potato fashion, just like the ETC did in cellular respiration. But it's a little bit different, and it happens first. So this is what we call Act 1 of photosynthesis. It's the photosystems. You're going to see the electrons being transported. I'm going to make some ATP in the similar way that I made oxidative phosphorylation, except over here it's going to be called oxidative photophosphorylation. And I'm going to make some ATPs, and I'm also going to carry some of these high energy electrons on a cousin carrier in a DPH. And you can think of that P as photosynthesis. It's not what it stands for, but it will just help you remember that. And then we're going to have this another cycle, and we call this the Calvin cycle. And the Calvin cycle is going to look similar to Krebs cycle. Not exactly the same in any stretch of the imagination, but it's going to be a similar mirror type of chemical reaction. And we're going to make glucose. So, just like cellular respiration was a redox reaction, right? A reduction oxidation process. Photosynthesis is a redox process, except water is going to be oxidized and CO2 is going to be reduced. So remember Leo. Remember Leo the lion goes grr, right? This is loss of electrons is oxidation. And if you can remember that, then you know what reduction is. Oxidation and reduction always go together. And remember our cheat, right? Wherever the electron goes, the hydrogens follow. So you can kind of see what's oxidized and reduced by looking at what happens to the hydrogens. So look here. Here's CO2. Where's the only thing that has a C on the product side of photosynthesis? Well, it's glucose. And look what was added here. I've got C, I've got O, C, I've got O. Here's some H's. So you're right. CO2 was reduced. It gained electrons. Well, if it's reduced, that means something else has to be oxidized. And look, water is oxidized. Water loses electrons, right? Water is oxidized, loss of electrons. And it goes from water to oxygen. All right? So we're going to have the reduction of CO2 into glucose, and we're going to have the oxidation of water into oxygen. The oxidation part happens first. The reduction part happens second. So here we have the two basic parts. I've been calling this Act 1 of photosynthesis, and this is Act 2. All right? So Act 1 is the light-dependent reaction. We used to call this the light reaction, and I still do sometimes, just out of habit, call it the light reaction because it is light-dependent. You have to have light, all right? The second part is called, it has three names, and you have to know all three. I'm so sorry. It's called the light-independent reaction. Oh, can't spell pendant. Pendant reaction. It's also called the dark reaction. Even though it doesn't have to happen in the dark, it just doesn't need light. And then the other name for it is the Calvin cycle. I'm so sorry, but you need to know all three of those names, okay? So to make things simple, a lot of times you'll hear me say light reaction and dark reaction, And that's not really, I should say light dependent reaction and light independent reaction. But sometimes I slip up and just call it the light and dark reaction, okay? So here's what's going to happen. We're going to start with light in the light reaction. The light is going to, the action is going to be in the chloroplast on the thylakoid membranes, all right? And what we're going to see is we're going to see ADP, inorganic phosphate, and the oxidized version of the electron carrier, NADP+, are going to be my reactants. I'm going to see oxygen coming off right away, and I'm going to make some ATPs and some NADPH. There are going to be some high energy electrons that are going to be carried there. And then in the stroma of the chloroplast, right? The stroma of the chloroplast is where the dark reaction or Calvin cycle or the light independent reaction occurs. And this is where carbon dioxide is going to be reduced to form eventually glucose. So let's see, we're going to look very quickly at a preview of both of these, and then we're going to dive in deep, guys. We're going to follow the electrons, okay? So photosynthesis comes from the photo part of the light reaction or light dependent reaction, and the synthesis part comes from the Calvin cycle or the light independent or dark reaction. So basically what's going to happen in the light reaction, the first part, we're going to split water, and water is going to be split because I really need the hydrogens and the electrons. Those are the things that I'm going to need in photosynthesis. What's left over is the oxygen. And I'm just like, I just kind of spit that out, right? So that's going to be a waste product. These electrons are going to be carried on the NADP molecule to make NADPH. And as I said, this is kind of a cousin electron carrier to NAD from cellular respiration. I'll show you the structures in just a minute. And we're going to make some ATP from ADP, and we're going to, that process is going to be photophosphorylation. And it's going to be an electron transport chain again. We're going to see that in electron transporting, and we call these a light photosystem. We call it a photosystem, and we'll see those in just a minute. Then in the second part, which I'll lecture on later in part 2, we're going to take carbon dioxide using the ATP and then NADPH from the light reaction. Remember, these were electrons that I needed. And then we are going to fix the carbon dioxide. Fixing kind of means like it's going to be incorporating the CO2 or gluing the CO2 into some organic molecules that are going to end up being my sugar. So here is a cartoon of the general all over diagram of what's happening. Here's light reaction on the right, on the left. And here's the Calvin cycle or the dark reaction or the light independent reaction on the right. So light comes in. This is happening in the chloroplast. This whole thing is happening in the chloroplast, right? But these are happening on the thylakoids in the membrane of these grana. I'm going to make some ATPs and some NADPHs, and I'm going to use water in a really cool way, you're going to see. But what's left over of the water that I don't need is oxygen, and that's just going to be spit out as a waste product. And then I'm going to take carbon dioxide in through my stomata, and I am going to fix that. I'm going to fix the carbon dioxide into some molecules. I'm going to use these ATPs because this is a endergonic reaction for sure. So I need ATPs to fuel that, and then I'm going to need the electrons because I'm going to reduce a bunch of things to eventually make sugar. So let's talk about light for just a minute, okay? So this is the electromagnetic spectrum. And this is showing us all of light, light with a capital L, all right? So this is all electromagnetic radiation or light, all right? And we can see that very, very short wavelengths of light are called gamma rays, and we have very long wavelengths of light that are radio waves. The part of light that we see as humans with our eyes is a very tiny slice of the electromagnetic spectrum between 380 and 750 nanometers of light. This is the wavelengths of the light, and we can see my Roy G. Biv colors. Roy G. Biv, right? So this is red, orange, yellow, green, blue, indigo, violet. Except the Roy G. Biv is actually in the opposite order of the energy of the wavelength, because this is the longer wavelength and violet is the shorter, okay, wavelength. So the shorter wavelength is the higher energy, all right? And so it actually goes Roy G. Biv, okay, Roy G. Biv. This is, of course, is the part of the sun's energy that's going to be important in photosynthesis. So when we look at light coming in, all right, light is all of that light that we talked about, but the visible light, all those Roy G. Biv colors of visible light, we don't use all of them in photosynthesis. And that's a really interesting discussion that we can talk about sometimes when we talk about evolution. But a lot of the wavelengths of light are absorbed. So we talked about Roy G. Biv, right? The one wavelength of light that is not absorbed, that is reflected, is green light, right? And that is why we see plants as green. That is why we see chloroplast as green. That's why we see the pigment chlorophyll as green, because it is reflecting that green wavelength. It's not absorbing it, but it is absorbing all these other wavelengths of light, as we'll see, and that's where the energy comes from that drives photosynthesis. So let's talk a little bit about pigments, all right? A pigment is something that's going to absorb visible light, and different pigments absorb different wavelengths. Of course, these are wavelengths that are not absorbed. The wavelengths that are not absorbed are going to be reflected or transmitted, and we just said that leaves appear green because chlorophyll, which is a pigment, reflects green light. It does not absorb the green light. So in one of the labs that we're going to be doing for photosynthesis in AP biology, we're going to use a spectrophotometer. This is a meter that is going to look at the different wavelengths of light. And we can see here that if we look at the y-axis, we can see the absorption of light of chloroplast pigments. Well, there's going to be three main ones and then some other accessory ones. We're going to have chlorophyll A, chlorophyll B, and some accessory pigments called carotenoids. Look at what wavelength chlorophyll A, which is my favorite color, it's kind of a blue-green color, absorbs light. It's going to absorb light right at this wavelength, right? Which is not. Look at the green. See how it's not absorbing there? So that is, it's reflecting that green. Chlorophyll B is going to absorb light in this wavelength, okay, right about here. The carotenoids have kind of two peaks where they absorb. So we also have some secondary absorption here in the orange-red, but we absorb a lot of energy in the indigo and violet and blue regions, and we also absorb in the orange and red, not so much, well, none basically in the green. So as I said, chlorophyll A is the main photosynthetic pigment. We also have chlorophyll B, and we also have some carotenoids, which also absorb some excess light. Too much of this light, though, would damage the chlorophyll, and that's why we think we need these accessory pigments. So, guys, here is chlorophyll. Isn't it a cool molecule? And if you look at this little functional group right here, if it is CH3, it's chlorophyll A. If it's CHO, it's going to be chlorophyll B. And then there is this structure called a porphyrin ring. This is the light-absorbing head of the molecule, and in the center it's got a magnesium atom. Do you see these hydrocarbon chains there? Right? So I would expect that to be hydrophilic, and that's going to be embedded in the membrane of the thylakoid inside the chloroplast, right? So that hydrophilic part, so it's going to be sticking in there kind of like a little lollipop. So my husband and I were so excited a few years ago when we had solar panels installed on our house. And that just makes us really happy to feel that we are producing electricity for our home in a sustainable way. But anyway, how does a solar panel work? Well, a solar panel takes energy from the photons that are released by the sun. Okay, so here's my sun. And those photons are basically going to knock electrons off the material in the solar panel. And a lot of the solar panels are actually made of the element silicon. And so those electrons get knocked off by the energy of these photons, and they start a chain of electricity, right? That's what the flow of electrons is electricity. So that's how our solar panel works. You're going to see an echo of that when we talk about how a photosystem works in a leaf. So a photosystem is a light center complex associated with light harvesting complexes. All right, so it's a protein complex that has a reaction center in the middle, and then it has light harvesting complexes, a lot of which include pigments that kind of funnel the energy of the photons to the center of the reaction. So in the center of the reaction center, you're going to have a primary electron acceptor that is going to accept this electron that gets knocked off from the photon. And it's going to be actually from the photon is going to come in, and I'll do that in yellow. The photon is going to come in, it's going to knock an electron off the chlorophyll A. That's what's going to start this. And then this solar-powered transfer of an electron from the chlorophyll molecule to the primary electron acceptor is the first step in the chain of the light reaction, right? So here's what this is going to look like. All right, let's see where we are first. We're in the chloroplast. We're in the thylakoid, on the membrane of the thylakoid. Okay, and I have some pigments here, and some of these pigments can be accessory pigments. This one right here is important because this is the special chlorophyll molecule. And depending on whether it's photosystem 1 or 2, it's going to absorb energy at that wavelength. And what's going to basically happen, guys, is you're going to have a photon coming in. It's going to be the energy from that photon is going to be reflected by these different accessory pigments until it gets on the main chlorophyll A molecule. And an electron from the main chlorophyll molecule is going to come off. It's going to be knocked off, just like it did in my solar panels. And that's going to be passed to a primary acceptor of an electron that is going to start a path of electrons. I know this is kind of tiny, but this is going to help you see what's going to happen. Now, you should look at that and you should say, Mrs. Foyt, this looks very similar to the electron transport chain in a mitochondria, doesn't it? It really does, okay? And again, it's going to be this system of electron hot potatoes where these electrons are going to be passed through some different protein carriers. We're going to have reduction and oxidation of those. The energy of these high energy electrons are going to use to pump hydrogen protons into a concentrated area. And then those hydrogen protons are going to be able to go down their concentration gradient through the enzyme ATP synthase, and I'm going to make some ATPs. And you might say, Mrs. Foy, that's so similar to what happened in the mitochondria. And yes, it is, but you'll see there's some differences. So here is just the photosystem, all right? So these purple things are proteins, guys, and they're embedded in the membrane of the thylakoid, right? And if this was my peppermint patty, this part would be the chocolate part of my peppermint patty, and this part would be the white part of my peppermint patty, okay? So photon light comes in. It's going to be reflected and concentrated by these light harvesting complexes that include some different pigments until it comes resting on a special pair of chlorophyll A molecules, and an electron is going to get so excited that it's going to jump off, and it's going to land on this reaction center in the primary electron acceptor, and then that's going to start my electron hot potato. There are two photosystems in the thylakoid membrane, and this is going to be one of those things that confuses you, and I apologize right up front. They are named in a very weird way, okay? They're named photosystem 2, which happens first, and then it goes to photosystem 1. But photosystem 2 was discovered second, and so that's why they call it photosystem 2, even though it's the first thing that happens, all right? And it's the chlorophyll is going to really like to absorb at that 680 nanometer range of light. Photosystem 1 is going to be better at absorbing photons that have 700 nanometers of their wavelength. And again, this one happens second, but it was discovered first, and so that's why we use the Roman numeral 1. And so the reaction center chlorophyll of chlorophyll A in photosystem 1 is called P700 because that's the wavelength that it absorbs best. So this cartoon is basically showing us what's happening to the electron, okay? Let's look at this. So here we have a photon of light coming in from the sun. It is going to be bounced around until it hits this P680 chlorophyll molecule. And it's going to excite this molecule so much that an electron is going to go popping off. And this electron is a high energy electron, right? And it is going to be transferred through a system of electron transporters in the membrane of the thylakoid in the chloroplast of a plant cell. And it is going to make some ATPs as it is oxidizing and reducing these molecules that are in this membrane. It's going to make ATPs in a similar way that we saw before. It's going to involve chemiosmosis. Then another photon of light is going to kind of give this electron a booster shot, and it is going to excite it again at the wavelength of 700. And that reboosted electron is going to then eventually be carried onto NADPH. So this would be NADP, the oxidized version, and this would be NADPH, the reduced version. Okay? Now, maybe you are thinking to yourself, but Mrs. Foy, what about that lost electron back at 680? How can that be replaced? And that's a great question. because that's where water comes in. And I'll just cut to the chase here. What's going to happen is water in the light reaction is going to be split. The oxygen part of water, and by the way, this splitting with the energy of light is called photolysis of water. And the oxygen is going to be given off, is going to be given off as a waste product. So this is going to be given off as a waste product. Sorry, I didn't like that color. Lord of mercy. All right. And then the hydrogens, we have two things that we're interested in. We're going to split that hydrogen into a proton and an electron. The electron from the water, specifically from the hydrogen of the water, goes to replace the electron that got so excited it jumped off at the very beginning of the light reaction. And so that is how water is used. So again, this system is going to look vaguely familiar to you, okay? So this one is in the chloroplast. I've got my photosystem 2 and my photosystem 1. And then look, I have got cytochrome again. Remember cytochromes? I have plastocyanin, plastoquinone, right? And so I have these special molecules that are going to be accepting the electron and are going to be accepting the electron, and they are going to be passing that electron on. Okay, and everyone that accepts it is going to be reduced. When they pass it back on to their neighbor, that's going to be oxidized. Because the electron on the original chlorophyll jumps off, I need to split water, and the oxygen is going to be released as a waste product, but I'm going to take the electron and put it back on the chlorophyll so the system can happen again. And the hydrogen protons are just ubiquitous, right? They're everywhere around, but I'm going to make a concentration gradient of all of these hydrogen protons by using the energy of the high energy electron that got energized from the photon to pump the hydrogen proton into the lumen, or the white part of our peppermint patty in the thylakoid. All right. And so I can see this electron goes here. It gets re-upped, right? It gets excited again by this another photon of light. And eventually it's going to end up reducing NADP to NADPH. And so now what's going to happen? Well, this NADPH is going to go on to the Calvin cycle. Okay, so stay tuned for that one. We're going to see it again. But meanwhile, I need to make some ATPs. Well, look, our old friend ATP synthase, remember? And so the hydrogen protons are going to go down their concentration gradient, but this time down their concentration gradient means they're going from the lumen of the thylakoid out of the thylakoid. All right. And that is going to create enough. Remember, this little thing is a rotor protein. It rotates. And when these hydrogen protons go down, it turns this rotor, and that gives enough energy for an inorganic phosphate to be bonded to an ADP to make ATPs. And the ATP is going to be something else that's going to go to the Calvin cycle, right? And so in the light reaction, I have two products that I need. NADPH and ATP go to the Calvin cycle to make sugar. But I use up water right at the very beginning, and I have oxygen that is released. Sorry, that word was waste, right? As a waste product. So let's look just a little bit at NADH versus NADPH. So these are both cofactors that transport electrons. NADPH stands for nicotinamide adenine dinucleotide phosphate, and you do not need to memorize that. You just need to know it's NADPH. And again, I think of that P as photosynthesis. Of course, it does not stand for photosynthesis. It stands for phosphate, but that's okay. It'll help us remember it, right? And NADH is nicotinamide adenine dinucleotide, and this is what happens in cellular respiration, right? So that's the one we use, the cofactor we use to transport electrons in cellular respiration. And look at the difference here, right? So there's just going to be one phosphate group, functional group, that's different, right? So they're very, very similar molecules. So now we're going to talk about the different pathways that the electrons can flow. During the light reaction, there are two patterns of this electron hot potato. One is called cyclic and one is called linear. We're going to start with the linear first because this is the primary pathway that the electron flows. It involves both the photosystem 1 and 2, and it produces ATP and NADPH. All right, so here is the linear or non-cyclic electron flow. And this is a good review, right? So we'll get to see everything, right? So here, photon of light coming in. Here's my little accessory pigment molecules that are going to kind of bounce around this energy. It finally hits the special pair of chlorophyll A molecules. These are the P680. Chlorophylls, and the electron gets so excited it jumps off, and that starts my electron transport chain in the thylakoid membrane. But meanwhile, back at the ranch, I have to replace this electron that jumped off the chlorophyll A. That's where water comes in, right? So we photolysis, we photolyse that water molecule, and oxygen is released as a waste product. But the electron goes back to replace the one that was lost on the chlorophyll. So what's going to happen is the electron gets passed down this electron transport chain. I am going to pump some hydrogens into the lumen of the thylakoid. I'm going to make some ATPs that way. Then I get a second photon of light. Bing, bang, bang, bang. It's going to end up on the P700 chlorophyll A's. The electron jumps off, and then it gets eventually bonded to NADP. This is the enzyme NADP reductase. Doesn't that say exactly what it does, right? And then this is going to go to the dark reaction or Calvin cycle, okay? Do you see how this is in a line, right? Right? It's like a one-way street. Sometimes, though, the plant cell or the photosynthetic autotroph cell will use photosystem 1 and not photosystem 2. If it uses only photosystem 1, it just produces ATP but not NADPH. And remember, we need two things in the Calvin cycle. We need ATP and we need NADPH, but we actually need more ATP than we do NADPH. And so the cyclic electron flow pattern that I'm going to show you next produces a little more ATPs that are going to satisfy that very endergonic Calvin cycle. So here's the cyclic electron flow, okay? And you can see that, right? It's going in a circle. It's only using photosystem 1. And you can see that I'm only using, some of my electron transport chain, but I make some ATPs, all right? But I'm not making any NADPH, all right? I'm just using this. And there's a lot of scientists that believe that this cyclic electron flow kind of helps cool the photosynthesis. It's not hot and cool, but it's similar to an engine kind of idling. It kind of this high energy can be dangerous to the proteins that are in the photosystems. And so this is a way scientists think of just kind of producing some ATPs, but not letting the electron get its full linear flow. So some organisms, like those purple sulfur bacteria, only have a photosystem 1 and not a photosystem 2. And so we think that electron flow, the cyclic electron flow, we think that evolved before the linear flow. And we think that the cyclic flow may protect the cells from light-induced damage. So I want to take just a minute to talk about free radicals. You might have heard of free radicals. We know that free radicals are involved in aging and to a certain extent in cancer. So free radicals are unstable molecules that actually have an unpaired electrons. And because of that, they are very destructive. They are highly reactive, and they can basically steal electrons from other molecules. So they can steal electrons from, say, RNA and DNA, and they can steal electrons from lipids and proteins. And so we have to be careful of these. They are byproducts of lots of different reactions in cellular metabolism. But we have to be careful with these. We can't just let electrons go flying around willy-nilly. And so that's why scientists think this electron, the cyclic electron flow, may have evolved. So, big picture, guys, we're almost done. You're doing great. So here we go. Photosystem 2, photosystem 1. Look where you are, okay? You are in a chloroplast. You're in a chloroplast. This stroma is the jelly-like substance, and this thing right here is the thylakoid, all right? Here is the lumen. Lumen is a fancy word for the inside of the thylakoid, and here's the membrane. Here's the membrane of the thylakoid, right? So photosystem 2, photosystem 1, right? Here are my electron transport chain, right? That are embedded in here, and I can see that electrons are going to go this way. Hydrogens are going to be pumped using the energy of those electrons. I'm going to have a very high concentration of hydrogen protons in the lumen of my thylakoid, and then they are going to flow down their concentration gradient through ATP synthase, just exactly like they did in cellular respiration at the end of the electron transport chain. The electron, though, is going to be bonded to or carried on the NADP to make NADPH. And the ATP that is made from this photophosphorylation, right, because the energy is coming from the sun to basically pump those hydrogen protons and let them go down through the ATP synthase. Now I've got ATP and NADPH, and then in the next part of this lecture, we're going to talk about what happens in the Calvin cycle. So this is a comparison of chemiosmosis in a mitochondria and in a chloroplast. In both cases, we're going to use an electron transport chain that is going to use the energy from those electrons as they're kind of being oxidized and reduced, as they're going down, being passed electron hot potato style, to pump hydrogen protons, to make a high concentration of hydrogen protons in one part of that organelle. And then the hydrogen protons are going to be able to go down their concentration gradient from diffusion, and they're going to go through that turnstile of the ATP synthase protein, and it's going to make ATP. The dark area is where the hydrogen proton, the high concentration of the hydrogen protons would be found. It is in the inner membrane space of the mitochondria. Okay, so I would have in that space, I would have a very high concentration of hydrogen, which means that I would have a low pH. Now you've got to be careful about this because you're going to get asked a question. Make sure the question, you understand it. Is it talking about where is there a high hydrogen concentration, or is it asking about the pH? Because remember, they're opposite. A high hydrogen ion concentration is a low pH. So you could look at this also as a low pH, acidic pH. All right? In the chloroplast, where is the chemiosmosis? It is happening in the lumen of the thylakoid space, right? So it's happening in the lumen of the thylakoid. And if you think of the thylakoid as the peppermint patty, it's going to be the white part. So we have just finished part 1. I've got you to here, all right? In the next lecture, I'm going to talk about how we actually make sugars in photosynthesis and about some different types of photosynthesis that we have. All right? And I hope that's been helpful, and I will see you in class.